Hello, I'm Dr. Stefan Wallace, and I'm here today to talk about an introduction to immunology. Following this presentation, I will give a talk entitled Introduction to Vaccinology. So let's begin. One way to think of the immune system is to imagine your body like a tree. First, it has bark, which is like our skin, nose, and ear hairs and eyelashes, and mucous membranes. These represent surfaces barriers that keep stuff out. There are some large branches, some twigs, and some leaves, representing different parts and functions. In this presentation, I'll use this metaphor to help guide you through the basics. I won't be talking about every one of these leaves, as some of them go well beyond the basics, but I did want you to see the names of the various parts, even those we will not be talking about today, just so you start to recognize the names if you hear them in a presentation. To start off, we'll look at the first branch of the immune system, your innate responses. These responses include cells like macrophages, which are like a garbage disposal, eating up cells that don't belong and helping the body dispose of them. Another innate response involves neutrophils. These are like the support troops. They follow behind the cytokines, which are chemicals produced by the macrophages, and do the cleanup duty. A third type of cell involved in your innate responses are the natural killer cells, abbreviated NK. The innate response is the first line of defense in the body, starting with your skin. NK cells are only one example of all of the components of the innate response. Can this type of response protect against infection? No. The innate response does not protect against infection because the natural killer cells are activated only after cells are infected by a virus. Unfortunately, this type of immune response does not have immunological memory. That means if you are infected by a virus, natural killer cells will fight against the virus. However, next time you are infected with the same virus, natural killer cells will not remember that they had fought the virus before, specifically fought the same virus before. NK cells cause the infected cells to burst and be destroyed, just like popping a balloon with a pin. Now we'll take a look at the adaptive immune response. The adaptive immune response is another way of saying acquired immune response. Your body acquires its knowledge about different invading germs over your lifetime. This type of immune response is directed toward a specific invader, such as a bacteria or virus. For instance, you will specifically develop an immune response toward measles or toward polio. As compared to the innate response, where natural killer cells start fighting right when cells are infected, the adaptive immune response will take several days to become protective. Think about when you get a cold or the flu and you feel really lousy for a couple of days. And then you start to feel better around the third or fourth day. That's your adaptive response kicking in. And the big advantage is that this protection develops or is acquired throughout your life. It is very important, especially when it comes to vaccines, where the objective is to ideally develop a lifelong protection. The first part of the adaptive response is called humoral. Humoral is what most of us know as antibodies. Antibodies are proteins produced by the immune system's B cells, which can neutralize or stop the virus. They are shaped like the letter Y. The antibodies can also help eliminate the virus. The good news is that antibodies can prevent infection. The even better news is that they have immunological memory. The first time you are infected by a virus, you will develop antibodies against that virus. The next time you come in contact with that same virus, you will already have antibodies prepared to fight against that particular virus. This is what preventive vaccines try to mimic. By giving you a product that appears to the body as the virus, the immune system creates antibodies, and the immune system will already be prepared to fight the real virus if a person is exposed to real HIV, as an example. This slide is only showing HIV as one example, but antibodies work the same way for all invading pathogens. The first way that antibodies can prevent infection is called neutralization. At the bottom in the yellow is one of your cells. It has receptors on the surface. You can see how the Y-shaped antibodies attach to HIV, blocking it from being able to attach to the receptors on the cell, thus neutralizing HIV and protecting the cell from being infected. Neutralizing antibodies are able to work independently to interfere with HIV, and they don't need help from any other parts of the immune system. In vaccine trials done to date, we have seen only weak neutralizing antibody production, which has not been strong enough to successfully neutralize HIV and keep it from infecting cells. The second way that antibodies prevent infection is by binding to HIV. Unlike neutralizing antibodies that can act by themselves, binding antibodies serve like a flag on HIV, which can call it to the attention of third parties. These third parties, the other components of the immune system, are able to recognize the invader once it is flagged and can be called into action. As an example, one way that binding antibodies can work is through opsonization, pulling the invader into the other cells that are able to break it down and eliminate it. Another way to think about this is to think of HIV as a piece of plain, dry toast. Think of the binding antibody as butter, which attaches to the toast and makes it taste better, making it more desirable. Now the other parts of the immune system will see this buttered toast and think, wow, that looks tasty. I'm going to eat that up. Another example of how binding antibodies work is ADCC, or antibody-dependent cellular cytotoxicity. In this example, the third party that is called into action is the NK cells. Now remember, the NK cells don't have any memory, so they don't recognize HIV by itself. But with the antibody bound on it and acting like a lookout, the NK cells can be called into action as killers. In summary, antibodies attach to the virus at sites that are used by the virus for entry into cells. Neutralizing antibodies can work alone to block a virus from entering cells. Vaccines designed to elicit neutralizing antibodies against HIV have not worked very well in trials so far. Recent discoveries of several broadly neutralizing antibodies are very exciting, and designing a vaccine to produce these antibodies is underway. Binding antibodies can attach to HIV and call other parts of the immune system into action to help destroy it. The second part of the adaptive response is the cellular response. The cellular response includes two main types of cells, often called T cells for short. These are the CD4 cells, also called helper T cell lymphocytes, and the CD8 cells, also called cytotoxic L lymphocytes, or CTLs for short. The cellular response cannot prevent infection, similar to NK cells. It gets activated only after cells are infected. The good news is that, different from the NK cells, this immune response has an immunological memory that can be trained by vaccines. This type of immune response may be able to clear an established infection. CD4s are helpers. Like a person with a megaphone, they help the various parts of the immune system communicate more efficiently. CD8s are the killers, just like this shark. What are the functions of T cells? They are essentially doing surveillance on your body. T cells will check other cells of the body to see if they are infected or look abnormal. And if they are, they will destroy them. This response is not only directed against infected cells, but also against abnormal cells. For instance, cancer cells. T cells will recognize a cell that doesn't look right, bind to that cell, and send the cell a message to self-destruct. The CD8 T cells are already programmed to work as the killers, but scientists are learning that they may also work more effectively if they see a binding antibody. Another example of how T cells work is called ADCVI, or antibody-dependent cell-mediated viral inhibition. Similar to ADCC that I explained a few minutes ago, having the antibody bound to HIV can also serve as a lookout or flag that helps the CD8 killers work better, making them even more successful killers. You may also see the acronym ADCP, antibody-dependent cell-mediated phagocytosis, which gets more specific to the type of cell that's doing the killing. Cellular, cytotoxic T lymphocytes, CTLs, or CD8s, and helper T cell lymphocytes, CD4s. In summary, the cellular response of the adaptive response cannot prevent infection. T cells are activated when cells become infected. T cells can eradicate an established infection. T cells do have immunological memory. They will remember a prior occurrence or infection. T cells can be primed by vaccination. Now we will transition to Introduction to Vaccinology. Preventive vaccines have been used for decades around the world and are most commonly found or given to children. Preventive vaccines are safe when manufactured and used properly. are a cost-effective means compared to treatment. And through the use of preventive vaccines, we've been able to eliminate smallpox worldwide. And in 2008 and 2009 and 10, we saw an approval for an HPV, or human papillomavirus vaccine, for girls and young women, as well as boys and young men. For this particular slide, I just want to point out how long it can take to develop a vaccine, even after we know what the cause of the disease is. While the time is especially frustrating with HIV because so many lives are being lost, we are actually on par with the timeline seen for other diseases. Also, there are no vaccines for any other retroviruses. And while it may take time, once we have them, vaccines have significantly reduced or eliminated a number of deadly infectious diseases. We hope one day to be able to add HIV to this list. Significant decreases are seen in the far right column. Measles, diphtheria, mumps, pertussis, smallpox, rubella. These are examples of the kinds of viruses that we've been able to eliminate or significantly reduce through the use of vaccines. But an HIV vaccine is more challenging. There are no human examples of someone clearing an HIV infection naturally. The closest example we have is the group of people known as long-term non-progressors, also known as elite controllers. These are people who are still infected with HIV, but who are able to control that infection and maintain a low viral load, often without the need for antiretroviral therapy. The one group of people who do have natural protective immunity are those who have a genetic mutation to the delta 35 region of the CCR5 receptor on their cells. You'll recall from the immunology presentation that those receptors are where HIV is able to bind onto the cell to cause an infection. For those who have the genetic mutation, their CCR5 has changed such that HIV is not able to attach to it. It's like trying to put a key into the wrong lock. It is unable to fit, and therefore it cannot infect the cell. This mutation is thought to have evolved from the Black Death in the 14th century, and it is most common among people of Northern European ancestry. People who have inherited a copy of this gene mutation from one parent typically have a much slower progression from HIV infection to AIDS, while people who have two copies inherited from both parents have more complete protection. The mutation can be found in approximately 10% of Europeans, but it is rare in Africans and Asians. The immune response we need must work for mucosal exposures, infant exposures via breastfeeding or through the birth process, and ideally in the blood for those who are infected through IV drug use. It must be a response in all the right places and at the right time. A vaccine is a substance that creates an immune response in the person's body so that if you encounter a similar substance or invader, In the future, your body will already recognize it and know how to fight it off. The vaccine causes the immune system to respond by looking as much like the invader as possible without causing disease itself. So, how does a vaccine actually work? Vaccines work by teaching your body to recognize the virus, sounding the alarm to activate your immune system, instructing your fighter cells and proteins to go into action, with the goal that the virus will be killed or controlled. So how do we make vaccines? Well, when you're making a vaccine, you don't want to give the person the actual virus or pathogen. This would just make the person sick. So you have to do something creative. Live attenuated. This means that the pathogen or virus is real, alive, but weakened. The Sabin polio vaccine works like this. Whole inactivated. This means the whole virus is used and it is killed or disabled in some way, usually with heat or chemicals. The Salk polio vaccine works this way. With these two traditional methods, manufacture is tricky because you have to be 100% certain that you've killed or weakened the virus just right. An unfortunate incident like this with the polio vaccine in the 1960s, known as the Cutter incident, and some people actually were infected with polio because of getting vaccinated due to a manufacturing issue. Up until now, researchers have not been willing to accept this risk with HIV. So these methods have not been used in human studies for preventive HIV vaccines. This slide demonstrates the many possible designs for creating any vaccine. Live attenuated has not been used with HIV, as there are safety concerns about using live HIV even if it is weakened. There have been similar concerns about using whole inactivated HIV. The first trial using this strategy launched in Canada in 2012 in people living with HIV, and there have been recent reports that it is moving forward to a phase 2 trial. With that vaccine, the HIV was inactivated in 2 ways, using chemicals and radiation to kill it. Aside from these 2 designs, the HIV Vaccine Trials Network has run clinical trials on most of these other designs. And as a note, virus-like particles shown at the bottom is the design like for Gardasil, the new HPV vaccine made by Merck. Perhaps one vaccine will not be sufficient to trigger both antibody and T-cell responses, and we will need a combination of products to attack the virus, similar to combination ARV therapy used to treat people who are living with HIV. In this scenario, one vaccine is given to prime the immune system in 1 or more doses, and a second vaccine is used at a later time point in 1 or more doses to support or augment the immune response. You can think of it like painting a wall in your house. You wouldn't want to just slap a coat of paint on the wall. First, you would prepare the wall by patching the cracks and smoothing any rough spots. Then you would apply a coat of primer paint. And then you would do 1 or more coats of your chosen color for the finished product. Similarly, the first vaccine primes the immune system to wake up. We're doing something here. And the second vaccine boosts the immune response. Several studies currently underway are looking at various combinations and prime boost strategies. One of the challenges researchers must figure out is which products work better as the prime and which work better as the boost. So far, it seems DNA vaccines seem to do better as a prime. And following the success of the TIE trial, also known as RV144, there are many new studies being designed to use a protein boost. So we're going to dig more into now designing HIV vaccines. Just in case you were wondering why this research is taking so long, take a minute to consider this list of all of the different elements involved in developing a vaccine. Each of these variables has to be tested to find the best combination. Even if you don't fully understand each of these terms, you can see just how many different options there are and how time-consuming it is to try to figure out the best choices. Vaccine modality basically means which method of vaccine design will you use. Whole killed, attenuated DNA, viral vectors are some examples. Genes represent the bits of HIV that you will include in the vaccine. Adjuvants, which one will you use? Will you use one at all? I'll talk a little bit more about that in a moment. Dose, how much vaccine should go in each injection? How many injections does a person need to get? Route, where is the vaccine given? Under the skin, which is intradermal, into the muscle, intramuscular, or other routes. And the timing, how much time should there be between injections? Do you need a booster shot at 1 year, at 5 years? And administration, will it be given with a needle, with a bioinjector device that does not have a needle, and instead gives the vaccine through the pores in your skin? Will you use electroporation? These are some of the elements that need to be determined, and the combinations are limitless. This is an image of the viral structure of HIV. You will notice the proteins, and each of these proteins has a different type. The GP120, GP represents glycoprotein. The membrane or the envelope, the matrix proteins, the nucleus or genes, the core proteins. Unfortunately, it seems like the one thing many people remember from their junior high biology class is the old ways of making vaccines, like live attenuated or whole killed methods I mentioned earlier, was to use actual pieces of the germ. This creates the misperception that preventive HIV vaccines might actually cause a person to get HIV. With new technologies and genetic engineering, this diagram is useful to explain how scientists use just pieces of the virus to make copies, synthetic copies, man-made copies, for use in study vaccines. The pieces that are used in the vaccine are called the HIV inserts. We can speak with confidence and say that it is not possible to get infected with HIV from study vaccines because we do not use any real HIV, only synthetic copies of pieces from HIV. If you think about it this way, if you have an eyelash, a tooth, and a fingernail can you make a whole human being? If you have a tire, a steering wheel, and a windshield wiper, can you construct a whole car? No. These parts are not capable of combining themselves into the whole person or whole car. These parts don't have the ability to create all of the other missing parts. In the same way, using just some of the genes or the envelope proteins from HIV cannot create a whole virus that is capable of causing infection. And thinking about HIV inserts, the idea behind HIV inserts is to show your body a piece of HIV because it doesn't need to see the whole virus in order to recognize it. In the same way, you know that slices of ham or turkey, peanut butter and jelly, and lettuce, slices of cheese, tomatoes, etc., are components of a sandwich even when you see them individually. But which of these pieces make the best sandwich? I'm sure many people could have a lively debate about that, just like scientists debate about which are the best pieces of HIV to use in a vaccine. So what carries the HIV inserts? The carriers of the HIV inserts are called vectors. A vector is a carrier that delivers the HIV inserts into your body's immune system. There are many different kinds of vectors, just like you can choose between wheat bread, a croissant, a panini, or a hamburger bun. Lots of variety and diversity. So making the vaccine or the sandwich, what if your vaccine needs a little something extra to give it more strength? What could you add? Adjuvants. I mentioned earlier that I would come back to this. You would add an adjuvant. You can think of them as the condiments you might use to give your sandwich or your meal some kick. You might add mayonnaise, mustard, ketchup, horseradish, and so on to your sandwich. Or you might add hot sauce, Tabasco sauce, or something to your meal. Adjuvants are products that are used to boost the body's reaction to the vaccine and give it that extra kick. Many licensed vaccines use adjuvants. One type of adjuvant that is used in Europe for many licensed vaccines is called MF59, which the HIV Vaccine Trials Network has tested in several studies. There are lots of different kinds of adjuvants. So, how exactly might an HIV vaccine work? There are several possible ways that scientists believe a preventive vaccine could work. If you think about the immunology presentation that I covered briefly before, the first way a vaccine might work is by triggering a strong antibody response. That would be the gold medal standard and completely prevent infection. But a vaccine that could trigger a really strong T-cell response might be able to combat an infection after it's already established and clear it from the body to prevent disease, just like we are able to fight back against a cold or the flu. This would be the silver medal approach. And the third idea is also related to T cells. Perhaps we couldn't completely fight off that infection, but only partially control it. This could result in slowing down the infection, making the time that it takes to go from initial HIV infection to developing AIDS stretch out even longer by helping to control viral load. This would be the bronze medal approach. And that could still be very important because we know that people with a lower viral load are less likely to transmit infection to others. And they are less likely to have opportunistic infections. There may be also benefits for the wider public in addition to the vaccinated individual. Herd immunity is the idea that even if only some people get vaccinated, making those people less likely to transmit HIV to others, then the wider community will be afforded some protection as a result. It may not be necessary to vaccinate every single person, but with fewer people transmitting, the larger public could still benefit. We are seeing an example of this in the US today, where we are losing our herd immunity to measles and pertussis, which is whooping cough. Since these diseases have not been a problem for many years, parents may think it is not important to vaccinate their children. In recent years, there has been a huge increase in numbers of measles and pertussis infections in adults and children. So now there are massive education campaigns underway to encourage people to vaccinate their children for measles and pertussis, as well as for adults to get booster shots. So what is the time frame for these immune responses that we're hoping to see with a vaccine? We know that HIV infection spreads very fast. This is why a vaccine is so crucial. We need to prepare the body before it encounters HIV, because what happens in the first 3 weeks of infection seems to set the course for the next 10 years. The virus replicates locally in the mucosa or your pink parts for a few days. And then it moves into the immune cells, which carry the pathogen or virus into the lymph nodes. This is supposed to be good for the body because this is where the immune system keeps the cells that are supposed to fight off the virus. Unfortunately, these are the same cells that the virus loves to eat or infect. So HIV goes on a frenzy and destroys the very thing that is meant to fight it. Within 10 days, HIV spreads rapidly throughout the body. Most of our immune cells are in our gut. This makes sense since most of the foreign substances that we take into our bodies come through the food that we eat. This is where many lymph nodes are found, and this is where the virus seems to hide. One particular type of immune cell, called the naive memory T cell, is reduced by 80% within 2 weeks. We hope that an effective vaccine will train the immune system to react faster, disrupting this timeline and slowing down the damage that HIV can do. The graph on this slide shows the potential impact of an HIV vaccine in preventing new HIV infections between 2015 and 2030. I want to draw your attention to this side of the slide, which is a summation of all of this over here. And to say, there are 3 scenarios that we're looking at based on this modeling exercise that some colleagues did. And that is if we had a vaccine that showed 30% efficacy or effectiveness, and we were able to get it into 20% of the population, we could avert approximately 5.5 million new cases, or we could prevent about 5.5 million new cases between 2015 and 2030. If a vaccine was 50% efficacious and we got it in about 30% of the population, then we could estimate preventing about 17 million new cases of HIV in that same time period. And a more ideal scenario would be 70% efficacy with 40% coverage, which would prevent about 28 million new cases in that same time frame. So this modeling exercise clearly demonstrates that even if a vaccine works a little bit and with not so good coverage, that it could still play a massive role in impacting the epidemic. On a global scale, there are significant implications for monoclonal antibodies and vaccine development for multiple HIV clades or types with significant variability. In some cases, over 30% of the genome is different. I will say again, genetic diversity in HIV is massive. So the question is, can a vaccine be developed against subtype clade B in North America that will also work for people in South Africa or Thailand, where the subtypes of HIV are different? For any of us, our genetic differences represent about 0.1%. 0.1%. That's about the approximation of our genetic differences. However, as I mentioned, for HIV, the differences in the genomes or the genetics are about 10 to 30%. So that is massive. So getting an HIV vaccine that will work across all of these different subtypes is important, an important consideration in this process. So trying new ideas to get there. And one new idea is called a mosaic. The mosaic uses a strategy by teaching your body to recognize HIV inserts across different subtypes of HIV found around the world. And why is that important? Well, the slide that I just showed you prior showed the genetic diversity of HIV at 10 to 30%. So again, thinking about a vaccine that would work across this diversity and work across people in multiple places around the world is critically important. This particular type of idea of the mosaic is being tested in two studies. The Imbokodo study, also known as HPTN 705, HPX 2008, in several countries in sub-Saharan Africa, as well as the Mosaico study, HPTN 706, HPX 3002, which is being tested in the US, Mexico, Peru, Brazil, Argentina, Spain, Italy, and Poland. Now, mosaics, you know, when most of us may think about this term, many of us may think about art, but we're not talking about art in this context. We're using the term in a different way. With the mosaic HIV insert, the idea is that the genetic material is the chain of amino acids. You'll see the beads here. Coded by the mosaic will represent the most common features of HIV that your body might be exposed to naturally, you know, just out in the world. Thus, it may better represent many versions of natural HIV proteins rather than any singular natural version. I will say this differently. the way that these inserts are connected and pieced together, they're connected and pieced together to represent different HIV subtypes, again, that are found in different places around the world. So with the goal of being able to construct or put together an HIV insert or a vaccine that would represent being able to work and potentially be protective against all of these different subtypes if a person were to come in contact with them. And again, I will remind that, you know, even with this particular design type, that people cannot get HIV from these small pieces. And we've seen results in early stage studies in laboratory settings, as well as in small animals and then larger animal studies, as well as in early human studies that showed some promise to this idea. So that is why it's being tested in this way now. The Mosaic sequences are made up of the most common pieces of protein found at each position in the chain. So the chain is the chain of proteins that are included in the HIV when we talk about the HIV insert. So we have to find the most common pieces of the protein at each position in the chain when many versions of naturally occurring proteins are compared. So on the right of this slide, you will see 6 chains of proteins that are taken from different people who are living with HIV. If you look at position 1 here, you will see that blue is the most common and red is the second most common. So we will put blue and red in our positions here to design our Mosaic vaccine. And you do the same to find the commonality in each position on the chain. But looking at the color alone is what scientists refer to as the consensus. The thing that makes mosaics unique is that the shape also matters. Proteins usually look like squiggly drawings, as I'm going to show in a picture in a minute. The strings of amino acids that make up each protein fold up in very precise ways. So not only do we have to get the placement correct in terms of the positions, we need to get the shapes correct so that each protein in the string will be formed properly. So in our example here, it isn't just red and blue in the first position, it's red and blue triangles that are important. So it's not red and blue ovals in the first position here, it's the red and blue triangles. So it's the shape, it's the color in this example that are important. And to cover the most common variations, as well as the second and third most common, you need to really construct, you need to really construct a few different mosaics. And 2 or 3 may be used together. A computer program determines the best sequence, combinations, and exactly how many mosaics should be constructed. Using this technique, the mosaic can teach the body's immune system to recognize and respond to the proteins that are most likely to be encountered in the natural HIV infection. And they will look like their proper shape, just as the body would be exposed to in a natural infection. You can see that the mosaics don't look anything like real HIV here. But because of their design, they can help the body be best prepared for what it might encounter if exposed. Another idea so as we have heard, we're also testing a new idea of using broadly neutralizing antibodies in a strategy known as antibody-mediated prevention, or AMP. You might also hear the term passive immunization. So I mentioned in a prior presentation about immunology about neutralizing antibodies, in that in prior studies, the neutralizing antibody function was weak. So we're testing this new idea that involves broadly neutralizing antibodies. Everyone who comes across any kind of bacteria or virus makes neutralizing antibodies, but not all neutralizing antibodies are equal. Some, as I mentioned before in prior studies that have been tested, were wimpy, like the Superman on the left, only recognizing and fighting back against one particular strain or one particular pathogen. The exciting ones, the ones we're really, really excited about that we're looking at now, are broadly neutralizing in that they're able to recognize and fight back against many strains of HIV and different viruses. So as I mentioned earlier, the antibody that we'll be testing, I'm sorry, I didn't mention earlier. The antibody that we have been testing, we've been testing quite a few actually, but I want to call attention to one specifically, which is VRC01, was able to recognize and kill 91% of different samples it was tested against, representing many different strains of HIV. So this is literally the difference between this weak Superman and the strong Superman in terms of how many different kinds of viruses the antibodies can fight against and neutralize. And there are lots more antibodies that we're looking at. This picture just gives an idea of how many more broadly neutralizing antibodies there are that have been discovered in the past few years. And there are even more that are not shown here. The idea here is that we will be seeing in future studies that if you can block more HIV by covering different places rather, HIV uses to bind cells, you can increase the potency of a potential vaccine. So this is a really important slide, and it just really highlights the fact that there are different antibodies that we are looking at in different studies in order to determine other ways we might be able to neutralize HIV, with the goal of ultimately developing a vaccine that mimics this. And the take-home messages here are simply the antibody-mediated prevention angle, or AMP, uses broadly neutralizing antibodies to see if they can protect against HIV infection. This is currently being tested at various studies in different phases of research. And the Mosaic or Mosaico concept, a way of teaching your body to recognize commonly found HIV proteins that are used as an HIV insert, being tested in 2 large-scale studies that I referenced before. Thank you to everyone who contributed to putting together this presentation, and to our funders at the National Institute of Allergy and Infectious Diseases.